We are going to hold a webinar devoted to the knowledge commercialization from European Molecular Biology Laboratory. And we host it together with the technology transfer team from EMBL and representatives of the Innovation Authority. So we will start today from opening that will be delivered by Dr. Aviv Ze'evi, Deputy CEO of the Innovation Authority. Then we will go to the technology transfer team from EMBL, Dr. Julia Dieter and Dr. Jürgen Bauer. Then I will tell you several words about the specific program that is designated to support the Israeli companies that want to commercialize knowledge and technologies from abroad. Then we have a special guest today, Dr. Kasif Sadiq, founder and the CEO of the Dinov AI company. Actually, that's a great example of how the knowledge and technology from EMBL may be commercialized in Israel. And then we have two great research teams from EMBL that will present their projects, like examples of the technologies that are developed in EMBL. Okay, so regarding questions and answers, there is a Q&A section in Zoom. You may write your questions here, and during the webinar, we will answer question. Okay, so Aviv, I will be happy if you start. I think everybody will be happy.
Thank you very much, Rami, and thank you everybody for joining us. I can see that we have 45 participants right now in this webinar, and I'm really happy that we have our German colleagues here with us. And the main aim, as I see for this webinar, is to expose the options of EMBL to create new knowledge, and we hope that this knowledge can be a source for innovation for the Israeli industry. We have quite a few, I would say, funding tools which are directed for this kind of cooperation. The main one is the tech transfer from abroad, which is aimed to allow to the Israeli industry to import new knowledge from outside of the Israeli academia, with the hope that it will be a new source for innovation and multidisciplinary. So I would just, since we have our German colleagues and friends who have joined us today, I just want to say a few words about why are we targeting this kind of cooperation with, I would say, one of the leading European R&D infrastructures. So our main reason is the fact that Israel is a center of global tech industry as we see it. We are, I would say, if we are comparing ourselves to national hubs, we are ranked more or less number 3 in the number of companies which are able to raise capital in certain areas, and we need new sources of knowledge in order to create these kind of companies. So the fact that we have so many companies which are eager to get new ideas, that's one of the reasons why we are now looking for them outside of Israel. So of course, we will embrace any cooperation with Israeli academia, but in many cases, we want the Israeli industry to look for this knowledge outside of Israel, with the hope that the company itself will be funded in Israel. So we see that the Israeli academia is fertile ground for talent and innovation, but we are just we are only taking part in some of the I would say of the options for knowledge. So if we are 5 out of the top 50, it still means that 45 others good academia and good R&D centers exist somewhere outside of Israel. So there are many opportunities available in the Israeli high-tech, and I think the Israeli high-tech is somehow diversifying and disrupting new markets, and that's what's really important for us. So we are focusing really on deep tech ventures, which are fueling the next wave of innovation, and that's why we are very we are looking for, especially for knowledge which is mission-oriented, and EMBL, which is focusing specifically on molecular biology, is one of the sources which is relevant for any, I would say, knowledge which is based on a clean tech and etc. So climate tech, sorry. So we are not only looking for physical components, but in many cases, the source of innovation on the biology side can be integrated in biosensors, which are part of the deep tech innovation that we are looking for. So I will not go further about why we are focusing on tech transfer import, but as you understand, EMBL is for sure a very good partner. And with this, Rami, we can continue with the webinar. Thank you very much.
Thank you for the opening remarks. Now we are going to the technology transfer team from EMBL. Dr. Julia Dieter, the floor is yours, please.
Yes, thanks a lot. Okay, so yeah, thanks a lot for the possibility to present here. My name is Julia Dieter, and I'm from EMBL, which is the commercial arm and tech transfer of the EMBL. And we are happy to present some of our assets and also presenting EMBL and EMBL in general to all of you. And yeah, in the following slides, I will guide you through some of these aspects. I'm joining this webinar together with Dr. Jürgen Bauer, who is the group leader of the business development group, where I'm in. And Jürgen is also the deputy managing director of EMBL. My background is in biotechnology, and yeah, as I indicated, I'm working at EMBL as a business development manager, handling different contracts and also supporting spin-offs. So first, I want to give a brief overview over EMBL, which is the European Molecular Biology Laboratory headquartered in Heidelberg in Germany. Which is central in this map here. And EMBL has 6 sites which are across Europe, and each site has a different focus. For example, as you can see in the top left, there's EMBL EBI, so the European Bioinformatics Institute, strong in bioinformatics as indicated here. Then we have the EMBL in Grenoble and Hamburg, which are primarily very strong in structural biology, and they also have beamlines at their sites, where they can do the corresponding analyses. We have EMBL Barcelona, which is the youngest site of EMBL, and EMBL Barcelona is really strong in tissue biology and modeling tissues or even small organs, organoid models, to test drugs or to model also developments in disease. Then we have the EMBL Rome, strong in epigenetics and neurobiology. And yeah, as I indicated, the headquarters, Heidelberg, strong also in life sciences, molecular biology in general, but also, for example, in imaging, where the imaging center has been built newly on site and where also a lot of research and improvements in imaging is going on. EMBL has operating programs, which are 5-year terms. And the current program is from molecules to ecosystems, including several transversal themes. So as you can see, it's a broad variety of topics where EMBL is really exploring the basics in life and molecular biology, involving, for example, human ecosystems, understanding the human body and its reaction to environmental cues, and on the effects of the environment on the human biology. Also, there is infection biology, exploring the detailed mechanisms of how pathogens, for example, enter the human body and how they interact with the body's biology. And, for example, data science, curating data, providing also really important resources on the internet, which are used worldwide by scientists. In addition, as a general structure for EMBL, it has 5 missions. As I indicated, of course, excellent research. This is where EMBL is really strong. And it performs fundamental research in molecular biology and the life sciences. And besides, EMBL is also really active in providing services. So this also involves academic institutions, but also industrial customers. And they approach EMBL where the services are performed, and EMBL is also really routined in that. Besides, EMBL is providing a vast amount of different training possibilities. It has also built a special building for it, which is the Advanced Training Center located in Heidelberg. And there are a lot of courses going on, really from covering the broad variety of all the different fields where life science or life scientists need to have expertise. For example, data analysis, programming, but also courses, of course, on microscopy, etc. And there's also a pillar for innovation and translation, tech transfer. This is where Emblem comes in, and I briefly want to also introduce Emblem to you. So Emblem is the abbreviation for Enterprise Management Technology Transfer. It's a fully owned GmbH, so fully owned by EMBL. It was established in 1999. And through all that time spent now, was really active, as you can see here, in spin-offs, in supporting spin-offs and creating spin-offs. The most prominent, maybe, which I could highlight to you here, is Cellzone, which is a spin-off from EMBL and was acquired by GSK. And from that, there is a really strong interaction and collaboration ongoing between GSK and EMBL. And it's a nice example of how this can thrive, because Cellzone is also still located on the EMBL campus, and the bonds with GSK are really strong, therefore. Besides this, EMBL also supports a public-private partnership, here in this case Open Targets, where several companies are active and collaborating with academic institutions to repurpose drug approaches and being active applying the latest state-of-the-art to achieve common goals. And also, maybe lastly to highlight in this slide, there's a strong collaboration ongoing based on the imaging center located in Heidelberg with Leica, for example, or other companies, where together there are developed new approaches in imaging with the scientists. In addition, we from Emblem use several routes, which began for doing tech transfer, and this should then result in some value creation from the technologies and the result and the research created by EMBL. For example, we have several licensing contracts. We are really active in this, also involving several partners from industry. As I indicated in the previous slide, there are also a number of startups being built from us, from EMBL and Emblem, of course, also creating many jobs and investments. I have also indicated the public-private partnerships and the collaborations ongoing with several industry companies, with leading companies in different fields, for example, imaging, but also in microfluidics, microbiology, etc. And yeah, maybe a brief overview. I won't go into detail into all these numbers, but just to show you the variety where Emblem is active. So I have indicated the spin-off companies and the licensing, but of course we also have a patenting arm, which covers all the processes regarding patents, for example, applications, deadline monitoring, and also fostering these patents and the copyrights. Our goal for this webinar would be to present our own assets, some of our assets, to also learn about the Israel Innovation Authority, and maybe also about the participants, and also to pave the way to start and to kick off new discussions, to maybe and hopefully have some collaborations in whichever form in the future. So once again, thanks a lot for the invitation to be part of this webinar, and I'm also really looking forward to the discussion. Thanks a lot.
Thank you very much, Julia. I just want to remind to those who have joined our webinar recently that there is we are close to 70 participants. There is a Q&A section. All the attendees are invited to write their questions in this section, and we will, the speakers will answer during the session. Okay, now I have 5 minutes for myself, so let's talk some money. Okay, I will tell you about the technology transfer program in the Innovation Authority. Just one moment, I will share my screen. Could you see it? I hope. Is okay?
Yes, we can see the slides. Yeah.
Great. Actually, the Innovation Authority has a special program to support the knowledge transfer and commercialization from abroad in Israel. Okay, there are several goals of this program: to validate knowledge, to ensure that the knowledge that was developed, the technology that was developed in the academia is okay for the company, may be used by the company in terms of the real economy, to adjust this technology to the specific needs of the company, and to implement the whole project in the company. These are the goals. Okay. Actually, we are focused on creating collaborations between academic partners and Israeli industrial partners. We want to close the gap between the academic institution and the needs of Israeli company. We want to put the technology from the academic shelf and transfer it to the company to make a maximum additional value. The foreign research institution, in our case, for example, EMBL, may be in the application of the Israeli company. The EMBL may be as a subcontractor, maybe put in the application as a subcontractor for the Israeli company. It means that we have all the communication between the authority and the Israeli company is directly between us. EMBL is a partner, but without direct communication with authority. The EMBL is a subcontractor for the Israeli company. That's the idea. As I have told, some money. Okay, let's talk money. The total budget that may be submitted to the authority for this certain research project, technology transfer project, is close to 850,000 euro, up to 2 years, and the authority finances up to 66% of the approved budget. So maximum grant is 560,000 euro. I mean, that's approximate numbers because we are operating in Israeli currency, in shekels, but here is for to illustrate the numbers. The part of the academic institution in the budget may come to 50%. It means that the part of the EMBL in the application may get to 425,000 euro. That these are the terms. You know, this program, technology transfer program from abroad, has got a lot of attention recently, and this year we have applications from the CERN in Schweiz, Switzerland, from Australia, Austria, Germany, Serbia, Switzerland, United States, and there are several more on the way. So I can say that the Israeli companies now much more aware of this instrument of opportunity to get knowledge and technology from abroad and to commercialize it in Israel. The application process actually is very simple. All you should do prior to application is to get, I mean, if you are a company, you should write a memorandum of understanding, a letter of intention with the academic institution, with EMBL. And in this memorandum, you should state the definition of the IP, the conditions of the commercialization, responsibilities of the parties. If your project is approved, I mean, this MOU should be changed by the written agreement, legally binding agreement. That's a preliminary term for activating your project. So the tasks that are eligible under the technology transfer programs: validation, adaptation of the technology, and training of the personnel that will be dealing with the specific knowledge or technology. I have mentioned that the application should be submitted by the Israeli company on a yearly basis, maximum 2 years. These are the documents that should be submitted with the application. All the documents of the forms are available on our website. If you have any questions regarding any of the aspects I have mentioned, please contact me. Here's my email, or contact directly to the authorities' email. That's all about the technology transfer program. Once again, if you have any questions, please write it in the Q&A section. And now I'm happy to invite Dr. Kashif Sadiq, founder and the CEO of Denov AI, the startup, the successful startup that was born as a result of technology transfer from EMBL in Israel. Dr. Kashif.
Thank you. Thanks for the introduction. It's a real pleasure to be here. So yeah, I'm really happy to share our story. Yeah.
Yes.
And it's in the normal mode, right? Okay, so great. So yeah, it's a real pleasure to share our story at Denovi. So Denovi is a de novo protein design company, and as you probably know from it's pretty famous at the moment. There's been a huge revolution in computational structural biology. In fact, this year's Nobel Prizes were awarded for that. So this is an incredibly hot area, which really promises to completely revolutionize the way biology and really life sciences are done in the future. So it's an extremely exciting area to be in. And, this kind of story really begins, a few years ago at EMBL, and then the transition to founding Denovi in Israel and building up a company there. So just to give you an overview, so proteins are really the workhorse of all living things, and they will impact pretty much every single sector out there, from health to agriculture, climate, and even energy. So being in a position to understand what proteins do, how they work, how they function, and being able to design them is really at the core of, in my opinion, one of the kind of fundamental innovative pillars of the 21st century. And that's again borne out by this revolution that's been happening in structural biology. So for decades, it was incredibly difficult to understand what the structure and the function of a protein was. You had to do very long, laborious experiments. And there was a long-standing question of whether you could kind of predict the structure of a protein just from understanding its components. That was called the protein structure prediction problem, protein folding problem. And recently that got, this year, got awarded the Nobel Prize, mainly because of now the very infamous, very extremely famous technology method called AlphaFold, but also some of the foundational work in protein design that was done by David Baker and colleagues in the USA. So that 50-year problem has to a large extent been solved in what was AlphaFold and many other technologies that have been spawned since then. So my background's actually in theoretical physics, and I came to biology. I did a PhD in theoretical biophysics, and then spent an academic career for about 15 years in computational biophysics. And then from there, I got into protein design, and actually specifically when I joined EMBL several years ago. And so we were actually one of the first people, alongside the great kind of collaboration between DeepMind, who created AlphaFold. EMBL was really heavily involved in partnering with DeepMind to take that technology and apply it to solve the structures of millions and millions of proteins known to humanity. And we similarly in the Heidelberg, so that was at EMBL EBI, and we at Heidelberg, we were one of the first groups actually in the group of Jan Corbel to actually take and build on AlphaFold to solve the inverse problem, which is of protein design. So not can you predict the structure of a protein from its sequence, but can you engineer a protein? Can you, if you know the design features that you want in your protein, can you figure out the sequence that will give you those exact design features? And that's exactly what we did. So we embedded that AlphaFold in a much larger algorithmic framework called Alpha Design. And we were able to show that we could design completely new proteins that did not exist in nature, but yet folded into stable three-dimensional structures, whether they were monomers or higher order, very complex proteins. And one of the key kind of applications of that was, well, now that we have this technology, we can actually design binders to target proteins. And that was extremely exciting because that opens up a whole array of different possible applications. So we patented that technology at EMBL, of course, through Emblem. That was an incredibly great thing to do for us, a great assistance and really rigorous process at Emblem. And then that was the basis to then license that technology into Denovi. So why did we choose Denovi? Why did we choose Israel? Well, so we were also excited by drug development generally, and we know that the process is incredibly slow. There's lots of failure, and we wanted to kind of apply in the first direction in a pharmaceutical direction. And so naturally at EMBL we were thinking in that direction, and that brought us into complete kind of overlap with what was going on in Israel, and namely through the Ion Labs partnership. So probably everyone in Israel is very familiar with that. But just to summarize, Ion Labs is a really amazing new venture, which brings together multiple different partners into this kind of unique alliance. So you have pharmaceutical partners, and you have funders, and you have the Israeli government, the Innovation Authority, and basically come together to solve a much bigger problem, which is essentially to found companies. So here you have Teva, Pfizer, AstraZeneca, and Merck, and they've come together to basically say there's a whole bunch of problems in the field that need to be solved. Can we figure out a way of solving this to create new companies that will solve this problem? And that's where Ion Labs was born. And that's what attracted us to this kind of model, because it was the first example of actual collaboration in the field, where these are not competitors, they're actually interested in solving the underlying problem. Sorry, excuse me. So that's kind of what brought us on to Ion Labs. And then the company is now really thriving. So we're really attacking the main problem of biologics discovery. So one thing is to design proteins that can bind to targets, and then another thing is actually to develop antibodies, which is a much harder problem. And actually, we're kind of on the path of cracking that. So we have, sorry, excuse me. So we actually have a process, an engine, which is able to completely design from scratch de novo antibodies and proteins. And rather than taking years and months to kind of do this drug development program, which is like preclinical, we are able to do that in a fraction of the time at a fraction of the cost and actually broaden the scope to many, many new diseases. And so we actually completely are trying to revolutionize the whole basis that preclinical drug development happens at. This is a really, really exciting place to be in. It's all possible because of the kind of, I would say, the underlying wisdom of how the tech transfer happened. So the story that I really want to share, I mean, apart from presenting the company and the kind of success we've had there, is really to say that the kind of underlying philosophy of how the tech transfer was done was really correct and really, really kind of insightful, because there's many a time, and I've heard many stories where, you know, the tech transfer process can be so kind of daunting that it kind of puts off young innovators like myself and my team to essentially make that jump from academia into building a new company. So what makes that easy is actually the philosophy that in the end, it's a win-win situation. And for there, I really have to say, like, the kind of the partnership and both the rigor, the fairness, and the kind of collaborative partnership between EMBL at EMBL and between the Ion Labs in kind of making that work really helped that to grow. And so, you know, there's all, you know, without going into details of all the different kind of structures that can exist in order to make these kind of partnerships, from equity to licensing to consultancy, that kind of magic formula is what tech transfer needs to come up with. And I'm very happy to say that did happen, and that's why we're thriving now. So we are, you know, very rapidly growing. We founded last year in late 2023. And we're already grown to a team of 6 people. And of course, our co-inventors at EMBL, Michael Jendrus and Jan Corbel, where the lab I was in, where we invented this, still stay on as consultants. And it's actually much more like, you know, you feel like part of that family. So like, you know, there's a huge overlap of not only professional interest, but also just this kind of personal colleagueship and commitment to kind of make this thing work. And that's why we're really excited to go forward in this kind of collaborative partnership. And so, yeah, but that's been, you know, kind of our story, which has been really exciting, and it continues. It's still early days, and we're growing from strength to strength. So yeah, I hope we have many more happy days ahead. So happy to share the story and happy to take questions.
Thank you very much.
I'll leave it there. Yeah, thank you.
You want to add something?
Oh, no, yeah, there was an extra slide, but just to say, you know, we're very excited by the idea that, you know, the technology that we are building will essentially address all of these areas. So, you know, I gave you the kind of sectors in the beginning of emerging areas in protein design, and really the future is bright in that sense. Like, there literally are so many different applications that we could do, and we're extremely excited to explore them in the future. And with that, I'll end. Thank you very much.
Great. Thank you very much. We hope that you are one of the most prominent examples of technology transfer from EMBL, but I hope not the last one. And from our current webinar, another collaborations will grow. Thank you very much once again. And now we are going to Sarah Shish, that will deliver a presentation about the opportunities in the framework of the European Horizon program. Sarah?
So thank you for giving me the opportunity to join this webinar between the Israel Innovation Authority and the EMBL. I am very happy to have the possibility to introduce to you briefly the Horizon Europe program. So hello everyone, I am Sarah. I am working at ISERD for Israel Europe Research and Innovation Directorate. ISERD belongs to the Israel Innovation Authority and serves as Israel's gateway to European research and development programs. As a national contact point for the Horizon program, I am your contact person for application to European grants in the health sector, with a specificity, European grants that require to build a consortium, which is a key aspect of the program. A bit of background, ISERD was established in 1996 to facilitate Israel's participation to the Horizon program. So what is this Horizon program? It is the European Union's flagship funding program for research and innovation. It has been running since 1984, and we are now on the program 2021-2027, which is the ninth iteration of the program. So this slide is quite complex, but here is the big picture. The program's budget for 2021-2027 is close to 100 billion euro. And the program is divided into 3 main sections defined by the European Commission as pillars. Under the pillar 2, the program is divided into thematic areas known as clusters. And I am responsible for cluster 1 into the pillar 2, cluster 1, which focuses on health. So now let's talk a bit more concrete about the program's challenges. There are 2 main challenges. The first one, to find the right European call for your entity, and the second one, to build a consortium. For those who are unfamiliar with the consortium, it is a collaborative project including at least 3 entities from 3 different countries, member or associated, with at least 1 entity coming from an EU member state. So all these countries, member and associated countries, participate to the program. And I am adding that the program is open to every legal entity. So for the cluster 1, for example, it can be startup, SMEs, large companies, hospitals, research institutes, universities, associations. Now, at ISERD, our main objective is to maximize the participation of Israeli entities to the Horizon Europe program. How? By disseminating to you the information regarding the opportunities that exist. It is the first point, finding fit calls for proposals, and by supporting you in your application to increase the success rates. Now, I want you to know that you can receive support and assistance from the Israel Innovation Authority. It is a track 37, project concept development support and red team. We are talking about reimbursement on consultancy fees, and travel expenses related to special events. You can also receive assistance from experts at the Israel Innovation Authority for project development or application review. But this support, this assistance is subject to budget availability. So please feel free to reach out to me, and we will check together if the resources are available. So that's all for the time which is allotted to me today. You are more than welcome to contact me or Nati, the Senior Director of the Life Sciences Department, for personalized assistance tailored to your entity. I would like to add that regarding the topics that are presented by EMBL today, that among the calls in the future Horizon Europe program, 2025 Horizon program, we have some calls related to antibiotic-resistant bacterial infections. My colleague Elinor, who is responsible of the cluster agriculture, you can also contact her for calls related to crop protection, sustainable agriculture. So please feel free to reach out to us. Thank you again, and good luck.
Thank you, Sarah. So just let's be clear, the EMBL may be partner in the applications of the Israeli companies in order to get financing in the framework of Horizon program. Actually, that was the bottom line. You are welcome to collaborate with EMBL and to apply for European grants as well through the ESERT. Thank you very much, Sarah. And now we are going to once again to representatives of EMBL research groups. Two more research will be presented as examples of the knowledge that may be transferred, for example, to Israeli companies. We will start from Dr. Elisabetta Casase. Yes?
Yes, hi.
Elisabetta?
Yeah, do you hear me?
Yeah.
Great. And I'm going to share my slides. Do you see the slides in correct mode?
Mm-hmm. Yes.
Okay. Yeah, first, yeah, thanks so much for including me in this event today. It's very interesting. I'm learning a lot. So what I'm going to present is something that I was involved in when I was at EMBL a few years back. Now I'm at ETH Zurich. And the inventions that we're going to show are all aimed at either fight multi-drug resistant bacteria or prevent antibiotic damage on the gut microbiome. So the overall goal is to refine and improve antibiotic therapies. And so this responds to two main problems. The first one being antimicrobial resistance, which I'm sure you're all aware of, which threatens all domains of modern medicine that are all based on antibiotics to be effective. And the other main issue is that new compound development is actually very slow and costly because of many reasons, mostly the hurdles that are there to bring new compounds to the market, and also finding new chemistry and new targets. And below you can see some numbers. These are old estimates for each pathogen in terms of deaths that are either associated or attributable to AMR. Now the estimates are a bit different, but still we're talking about almost 2 million deaths by 2050 due to antimicrobial resistance. So it's a major threat. And the other problem is that antibiotics, even those that are already in place, they come with a cost. So what happens is that they don't only target pathogens, but also commensals, so bacteria that we have in our body, in the gut or other body niches. And this leads to either short-term toxicity, which I'm sure you're also familiar. Whenever you take an antibiotic, there's side effects, especially on the gut, that you can experience, but also more long-term effects that are becoming clearer and clearer, such as dysbiosis or in general imbalances in the microbiome composition. And the other problem is that antibiotics and also non-antibiotic drugs, they exert a selective pressure in the gut on the commensals that then can become themselves a resistance reservoir, and they can spread these resistance elements to pathogens that can come along. And here you can see a study that was performed in the lab where I was and where all these solutions have been conceived. So the TPAS lab at the EMBL Heidelberg. And here you can see the impact in vitro of different antibiotics over here on gut microbiome components. And so all antibiotics have, as you can see, a cost. So this is the minimal inhibitory concentration. So what we thought of was to, for both of these problems, to devise therapies that would be more narrow spectrum, so that they would target more specifically pathogens, and also using combinations. They come with advantages, so that bypass the problems of new compound development. Because with combinations, what we can do is to repurpose drugs, antibiotics and non-antibiotics that are already approved, and therefore the translation to the clinics is much easier and much faster. What we are also presenting here is a method to screen for combinations, which of course are hard to test because of combinatorial explosion. So whenever you want to test combinations between few compounds, the number of combinations is very big, and this has costs in terms of time and money. And here we have a large-scale automated method that can do that in an effective way. And also allows for screening in a dose-resolved way, which is important for interactions, for synergies and antagonisms between drugs, because they are dose-dependent. So this was performed at EMBL in this automated robotic platform, and happy to take questions on this part later. So what specifically I did was to screen this number of combinations on different gram-positive bacteria and come with this final number of combinations, as I said, in a dose-resolved manner. So with these 4x4 checkerboards that can either be synergistic or antagonistic or neutral. And here's the quantification, which I won't describe too much, but again, I'm happy to take questions. And it's not on bioRxiv anymore. It was published years ago, last year actually, on Nature Microbiology. So first of all, with this method, so we could discover new synergistic combinations, which you can see here below. And I want to draw your attention on this one, which involves an antiplatelet drug, so a non-antibiotic drug that still can have an effect on bacteria, and it can synergize with antibiotics. And the pathogen that we were interested in was methicillin-resistant Staphylococcus aureus, and specifically strains that were resistant to antibiotics that for that bug are kind of last resort, so linezolid and tetracycline. And as I mentioned, this has this big advantage that these drugs are all approved singularly. Here you can see the data in vivo in a Galleria Mellonella infection model. And for this biosis, we applied a similar experimental setup where we asked the question whether non-antibiotic drugs, when combined with antibiotics, could act as antidotes. And so here you can see one example where dicumarol, which is again an anticoagulant, can rescue gut commensals from the collateral damage of macrolides, which are antibiotics. And this was validated in Bacteroidota, which are key gut commensals, very, very abundant, and was validated in mice, as you can see here. So the commercial potential is clear because the antibiotic market has this kind of numbers, but the main point I would say is the fact that the AMR is spreading, and there is the need for supportive government legislation. And as I said, for new compounds, this is tough. And this is the compound annual growth rate. To compare with existing solutions, so whereas antibiotics or non-antibiotics can be used alone and non-antibiotics are not traditionally used against bacteria, what we propose is combinations of both. So leveraging the chemical space that we already have. So synergistic effects are another point because, of course, you have additive and neutral effects, but synergies are more powerful. Antagonists are also important for clinical context to avoid them potentially. And until now, the combination testing was tough because of the experimental constraints that I mentioned. But with this pipeline, we can screen millions of combinations depending on the time scale. So those thousands of combinations that you've seen can be screened in 2-3 months tops. And so this is a very nice setup that can be used to resolve concentrations and strains also. And so this is the situations in terms of patents. So the first thing is the method. So this automated setup, and then that can lead, despite being so high throughput and so low volume and large scale, can still lead to in vivo valid interactions. Then we have this specific combinations of also non-antibiotic drugs in MRSA, again validated in moth larvae. And then the antidote concept, so non-antibiotics that rescue the gut microbiome, which was confirmed in mice. And the IP status is, as you can see here. So on this part, so I'm still a pure scientist. So it's great that Julia is here and she can take questions on this part. But PCT was filed in 2019 for this, 2023 for the combinations, and 2020 for the gut commensal antidotes. And here you can see more on this part. So also for the method, actually invention, there's combinations also there with non-antibiotic drugs, but on gram-negative bacteria that are also very interesting. And the development plan that we hope to go forward with is to move on from the inventions to the preclinical work which we already done to more clinical phases. And so we are potentially interested in out-licensing, collaboration, and technology evaluation programs. And with that, I'm happy to take questions. Thank you.
Thank you very much, Elisabetta. Actually, all the questions may be written in Q&A section, and we are, as panelists, we are able to answer them inside this section. Now, we are going to the second research spin-off correlates development of precision crop protection products as basis for sustainable agriculture, Dr. Mariana Vreto-Schultz and Dr. Jose Marquez.
So welcome to our company presentation. We are Cerelixis, and we develop high-value early-stage assets for the ag industry targeting crop productivity. One of the key challenges of modern agriculture is around food security. Climate change, new pests and diseases, and resistance to existing modes of action are all impacting yield and quality. At the same time, innovation in crop protection has slowed down and regulation has become stricter. This is where we Cerelixis come in and our proprietary technology, Crystal Direct. Our goal is to generate a pipeline of small molecules that improve crop productivity and out-license those to ag players. We start with well-validated biological targets, very quickly identify starting points for rational drug design, and equally quickly turn those initial leads into highly potent early-stage assets. At the same time, we put together de-risked data packages that will be attractive enough for an ag player to take them on and invest in late development, registration, and commercialization. A quick look at the founding team consisting of myself, Mariana Vreto-Schultz. I bring experience in the crop protection industry in both the R&D and the business worlds. And we also have Jose Marquez, who is a leading innovator of high throughput technologies. Let's have a little bit of a more detailed look at Crystal Direct. Crystal Direct is about automating the harvesting and data management of large libraries of protein ligand interactions, crystals, basically. And by doing that, the benefit is that we are able to screen a large number of small molecules against validated biological targets, very quickly identify active hits, and turn these active hits using rational drug design into highly potent molecules. The pathway that we have chosen for proof of principle for Cerelixis is the abscisic ABA pathway. It is highly conserved across plants, and it is crucial for the regulation of growth and stress responses in plants. If we activate the ABA pathway, then this leads to adaptation to drought, salinity, and cold conditions, as well as improved water use efficiency. And if we inhibit the ABA pathway, then this leads to improved homogeneous seed germination, accelerated seedling establishment, and improved primary root growth. All of this ultimately translates into higher yield and quality at harvest. Let's turn to have a look at our first assets by screening 2,000 small molecules and using Crystal Direct. We were able to rapidly identify active hits and turn those into nanomolar ABA inhibitors. At the moment, there are no commercially available ABA inhibitors available in the market registered for use in agriculture. We have also created a pipeline consisting of 2 chemical series that deactivate the ABA pathway. They are more advanced in development, and 2 chemical series that activate the ABA pathway that are earlier in the development pipeline. The market that we are targeting with our first assets is the plant growth regulators market. It is valued at $3.7 billion, and it has a very healthy growth of close to 9%. And we're looking at foliar and drip irrigation use in the field or in the greenhouse, and in the EU, this would fall under Regulation 1107. We are looking to reach the first value inflection point market, and we have clearly identified the key milestones to be able to achieve that. Starting with establishing efficiency in real conditions, we have identified a number of experienced CROs that would enable us to test for this in the field or in the greenhouse. We have put together a regulatory de-risking strategy using in vitro screening. We have put the strategy together with a validated CRO, and we are also aiming to improve the synthetic route for scale-up. This is work that we are just embarking on. So at this point, I would like to thank you and close with our ambition to become a leading company creating advanced solutions for the ag industry. Thank you very much for your attention.
Thank you very much, Mariana. I see there were several questions. We have one open question. Can IP be generated for the antibiotics products? Do you have an answer?
Yes, I think I answered it. I hope so in the chat. So the combination of the products is already the IP coming out of this project, and it was protected by the patents, as Elisabetta indicated in the slides. Yeah, so this is basically the treatment method or the approach as a new indication or a new treatment approach, basically, by combining these substances.
Great. Thank you very much, Julia. So actually, we are precisely on time. If you have, dear participants, if you have any additional questions, that's time to ask. We will wait for one more minute.
Yeah, so if I'm not wrong, I think there's a question that we have not addressed. Is the proprietary platform can be used for binding validation of small molecules and proteins in the biotech area? Was it done? Can you see that one?
I saw, but now I do not see. Actually, if you have question, if you have an answer, please.
I'm also, is it because it's not clear for who this question is. I guess it's for us.
So, dear listeners, please could you please write and send the question once again, so so we will know to whom it addressed. Oh, for Cerelix.
Okay. So just to make sure that I understand also the question correctly, I'll go ahead and answer, and if you're looking for something different, please let me know. So the proprietary platform, what it basically does is that you're able, because you start with a very well characterized target, biological target, and you are able in these protein small molecule co-crystals to screen for the small molecules that are able to truly well bind to the protein cavity, your receptor. Yeah? So by doing this initial screen, you can screen, let's say, 2,000 molecules the way we did it, and you can identify already from the very beginning hits, small molecules, that already have a pretty good activity because of the way the technology platform works. So you start then with quite an active hit, and you can, using rational drug design, really optimize it quite quickly to nanomolar level. Yeah. So yes, this is something that could generally be used in the biotech area, even in pharma. It's a concept in terms of the platform that has multiple uses. But in our case at Cerelixis, we would have the license, the exclusive license to use it for crop protection use. Does that address the question?
Can it also work for a complex of proteins? There's additional question.
Would it work for?
For a complex of proteins?
Complex of proteins.
Mm-hmm.
I'm not sure if Rosanne is here to answer that question because it's quite specific science.
Okay. Any other questions?
Yeah, we can get back to you on that one.
There was a question from Dorit Cohen. Thank you very much, Dorit.
Yeah, that's,
Okay, if there are no other questions, then we may finish our webinar. I want to thank once again our friends from EMBL. Thank you very much for organizing, for your help and collaboration during this during preparation work. The webinar is recorded and it will be available on our YouTube channel. So everybody who wants to share, please visit our YouTube channel and you may share the webinar. So thank you very much. If you have any additional questions, please address EMBL, Dr. Yulia Dieter directly, or address me or Sarah Shish if you have a question about ESART and collaboration with Horizon program. Thank you very much, Kashif, for presenting your company. Thank you very much, dear researchers. So hope we will see another applications for technology transfer grants. Thank you very much.